lecture, we will begin discussing Section 2 from Chapter 18, which focuses on the electron transport chain. You may recall from your intro biology courses that the ETC is the pathway which receives the electrons from NADH and FADH2 produced during the citric acid cycle in order to help power oxidative phosphorylation in making ATP. In class, we will be working on activity S34, where we will apply the content of this lecture in order to identify the important roles that coenzymes and prosthetic groups play in biochemistry, predict if ATP production is possible when coupled to particular reactions or processes, use Eknot values to predict the direction of reactions and the sequence of reactions, and to apply the principles of electron transport to a new situation. Additionally, we will spend a class meeting on a medical case study looking at the impact of certain diet fads on the ETC and human health. So overall, we will spend 2 class meetings working on this material. While Chapter 18 also covers oxidative phosphorylation, we will only be focusing on the electron transport chain for this lecture. There is a second lecture focused on oxidative phosphorylation. Additionally, Section 1 from Chapter 18 focuses on the structure and function of the mitochondria and may be a good review if you need to brush up on your basic mitochondrial knowledge. In order to prepare for this in-class activity, we will cover the following learning objectives in this video. After completing this unit, you should be able to meet each of these objectives, so pause here and read over them before proceeding with the lecture. The electron transport system is driven by the exergonic process of electrons being transferred down a redox potential, where that release in energy can be coupled to drive oxidative phosphorylation of ADP to make ATP. Each step in the ETC has increasing reduction potential so that the energy is released sequentially step by step through the pathway. Basically, each oxidized substrate's affinity for electrons increases with its standard reduction potential. So as electrons cascade down the ETC, we are continually releasing energy. We can calculate the energy released in a redox reaction by using the half reaction potentials and the Nernst equation for delta G. To determine the change in the reaction potential, you subtract the reaction potential for your electron donor, or the molecule that is being oxidized, from the reaction potential of the electron acceptor, or the molecule that is being reduced. For example, if we want to calculate the change in reduction potential for the overall transfer of electrons from NADH to O2, we would then calculate plus 0.82 for the reduction of O2, minus negative 0.32 for the oxidation of NAD+, to get the value of plus 1.136 for delta E. We can then calculate delta G using the Nernst equation, where N is equal to the number of electrons transferred, in this case 2 electrons, and F is a constant, and it describes the charge on 1 mole of electrons as 95,484.56 joules per volt mole. So in our example, delta G is equal to negative 2 times 95,484.56 times 1.136, or negative 217 kilojoules per mole. If you need a review on how to calculate the delta G from the change in a reduction potential, please see Section 3 from Chapter 14 in your textbook, or watch the supplemental video included on this unit in our course Moodle page. We will also get more practice using this calculation during our next in-class meeting. So as we just calculated, the transfer of electrons from NADH to O2 can release as much as negative 217 kilojoules per mole of energy. If we only require 30.5 kilojoules per mole of energy to make 1 molecule of ATP, how many ATP can we theoretically make? Well, 217 divided by 30.5 gives us a maximum potential of 7 molecules of ATP produced for every molecule of NADH that is oxidized in the ETC. However, we were told in our previous lecture that each NADH molecule only produces 2.5 molecules of ATP, meaning that our oxidative efficiency is only 35.7% effective. Where is all that excess energy going? We can discuss that in more detail during our second in-class activity on the ETC, so keep that thought in the back of your mind. Now that we know how to determine the energy released during the ETC, let's look at how this energy is released using sequential steps and multiple complexes. Overall, the ETC is comprised of 4 main multi-enzyme complexes and 2 smaller protein-based electron carriers. The complexes, as you can see here, are embedded within the inner mitochondrial membrane, and often there are multiple sets of these complexes all throughout this highly folded inner membrane so that we can rapidly consume any incoming NADH or FADH2. As NADH and FADH2 are created within the mitochondria during the TCA, they then immediately pass their electrons on to complex 1 and 2 respectively. The transport protein CoQ then transfer these electrons to complex 3, where cytochrome C picks up the electrons and transfers them to complex 4. Complex 4 then finishes off the pathway by transferring the electrons to oxygen, making water. During this electron transport, we then can couple the energy released through the reduction potential at each step to pump protons against their gradient from the mitochondrial matrix into the inner mitochondrial membrane space. As you can see, complex 1 and 3 both pump 4 protons, while complex 4 pumps a total of 2 protons, and complex 2 does not pump any protons at all. To see why we have differences in the number of protons pumped at each complex, we need to take a look at the ETC step by step. In complex 1, NADH transfers 2 electrons first to the coenzyme complex FMN within this enzyme multi-complex. Those electrons are then transferred to multiple iron-sulfur clusters and are eventually picked up by CoQ. While FMN can receive both electrons simultaneously from NADH, the iron-sulfur clusters can only transfer 1 electron at a time to CoQ. As these electrons are transferred through complex 1, we produce enough energy to pump 4 protons total into the intermembrane space. Do you recognize the metabolite succinate? I hope you do. Complex 2 actually contains the citric acid cycle enzyme succinate dehydrogenase. In that reaction, if you recall, electrons pass from succinate to a covalently bound molecule of FAD in the enzyme complex to make FADH2. Those electrons then pass immediately through iron-sulfur clusters and to cytochrome B560 and then eventually are picked up by CoQ. CoQ, which is lipid soluble, can then diffuse through the lipid bilayer of the mitochondria between complexes to transport electrons. As you may notice, there are no protons pumped from complex 2. This is due to the fact that this enzyme complex does not completely transverse the membrane, and this is one of the reasons that electrons transferred from FADH2 only produces 1.5 molecules of ATP for every molecule of FADH2 versus NADH's 2.5 molecules of ATP. Later on, when we look at oxidative phosphorylation, we will see how the number of hydrogens pumped per complex is used to calculate how much ATP is actually produced. In complex 3, electrons are picked up from the reduced CoQ and are transferred through 2 B-type cytochromes, 1 cytochrome C and 1 iron-sulfur cluster, and are eventually picked up by cytochrome C transport protein. Because CoQ is a 2-electron carrier, where cyto C can only accept 1 electron at a time, the ETC utilizes the Q cycle, where 1 electron is transferred through complex 3 to the transport cytochrome C, and the second electron cycles back to CoQ for 1 round and then is transferred to cytochrome C. This cycle also allows complex 3 to pump 4 protons from the matrix into the intermembrane space per electron pair. Complex 4 then picks up electrons from cytochrome C 1 at a time and catalyzes the 4-electron reduction of 1 oxygen molecule for every 4 reduced cytochrome Cs. This complex contains 4 redox centers: cytochrome A, cytochrome A3, copper B, and copper A centers. Cytochrome C oxidase has 2 proton translocating channels. The first protons from the matrix are used to form water upon the reduction of O2, and then the 4 electron reduction reactions translocates an additional 4 protons out of the matrix into the intermembrane space. Therefore, for each molecule of oxygen that we reduce, the matrix loses 8 positive charges. Again, we'll see why this matters when we talk about oxidative phosphorylation. Overall, our reaction at complex 4, we see that requires 4 protons, 1 molecule of oxygen, 4 reduced cytochrome Cs, and yields 4 oxidized cytochrome Cs, 2 molecules of water, and 4 protons that have been pumped into the intermembrane space. Unlike other pathways that we have reviewed, the electron transport chain is not regulated by inhibitors or activators. Instead, it operates solely based on the presence or absence of electron carriers NADH and FADH2, as well as the presence of oxygen. If these are present, it is running. If they are absent, it is off. However, there are molecules that can act as ETC inhibitors, and these molecules are often called toxins, as inhibiting the ETC can often result in tissue damage and death. Rotenone is a naturally occurring pesticide found in plant stems and leaves and is known to inhibit complex 1 of the ETC. Antimycin A is produced by the bacteria Streptomyces and inhibits complex 3. This molecule was first discovered in 1945 and was utilized by the American fishing industry as a chemical fish poison to help eradicate invasive fish species. While toxicity to humans from consuming contaminated water or fish is controversial and studies are ongoing, the EPA still allows use of this toxin under strict regulations. Cyanide is probably one of the most well-known ETC inhibitors, and it blocks complex 4 by binding to the iron within the enzyme, completely shutting down the electron transport chain. Cyanide is used in mining processes as a means of dissolving precious metals such as gold and silver out of ore. It can also be found in illegal fishing practices near coral reefs, as well as in pest control, particularly used to kill coyotes in the United States. Amytal is a class of barbiturates that can inhibit complex 1 and was once used to treat soldiers during World War II for shell shock, as well as studied as a potential truth serum. However, it has since been discontinued as the powerful sedation, cognitive impairment, and discoordination induced by the drug greatly reduced soldiers' usefulness in the field. Amytal is also the agent commonly used in physician-assisted suicide for terminally ill patients seeking death with dignity. One of the most famous and controversial cases in recent history was the death of 4-year-old Brittany Menard. In 2014, Brittany was diagnosed with a very aggressive, painful, and terminal form of brain cancer. At that point, unable to face 6 more months of pain, Brittany and her family became strong advocates for the legalization of assisted death through the Death with Dignity movement. Her and her recently married husband moved to Oregon, as assisted suicide was not yet legal in her home state of California. And on November 19th of 2014, she ended her life on her terms. At that time of her death, only 3 states offered some form of legal assisted suicide for terminally ill patients. And since her death, there are now 7 states total that allow death with dignity, in large part due to the advocacy efforts of Brittany's brave story and her family. Now that we have covered the steps of the ETC, learned how to calculate the energy released during electron transfer, and seeing how the ETC could be inhibited with various toxins, we will complete 2 in-class activities that will allow us to further explore this pathway in more depth.